Jai Bharat. Welcome to Advait Achievers. Pranam to all of you as we believe Vidyarthi Devo Bhava. So children, in this segment we are going to talk about the structure of human sperm. Right? We have already discussed the process of how the sperms are being formed. Now let us just see the structure of it. We all know that the human sperm which is there, it is haploid, formed by the reductional division of the germ cells, right? So it is haploid. Then it is a flagellated structure, right? And is motile. So it is haploid, flagellate and motile. Motile, it is able to move, right? So the sperm is actually made up of 4 parts, okay? It is made up of 4 parts, or it has 4 parts, we can say. It has a head, then a neck. The neck which is there is most of the time not visible because it is just slight, okay? Then it has a middle piece, and the tail, right? Now in case of head, when we talk about head, includes 2 things. It has the nucleus and the acrosome, right? We have talked about that acrosome has those sperm license, right, which is made up from the Golgi. You remember the bigger part of the Golgi was forming the acrosome, right? So just now let us take a look at the structure. Simultaneously we will draw and label and understand these different parts, right? So same. We said that the nucleus is getting condensed, it becomes pointed, oval and flat. And when we say the condensed nucleus, we need to remember what does that condensed nucleus has. And when I am drawing the nucleus, you can see I have put a slight depression over here, right? That was my main reason of erasing it. Okay, so this is the nucleus, as we said. This nucleus we said is condensed. So when I say a condensed nucleus means what was it having? It was having what? DNA and the basic proteins, right? So we'll add here DNA plus basic proteins, right? So this is the nucleus which has become condensed. On the nucleus part, okay, it has a small structure on it, which is we are calling it or which we call as the acrosome. So this small part we will be drawing, but it's a cover like of cap jaisa dikhega. Okay, so see, this is that acrosome. And remember, acrosome was formed from the Golgi. So this is acrosome. Into bracket we can add here to remember Golgi, that bigger part. Golgi breaks and it is having those bigger part. Then we said that the whole structure gets covered by a plasma membrane. So that also we will be showing. So, but I will be drawing it a little bigger because there are still 2 more things which I am supposed to add here. So I am drawing it a little bigger, but that is a little later I will be adding for you to better understand. Alright. So we will take it a little in the center so that becomes easy for you people to observe. Okay. Alright. So I have drawn it a little broader over here because I am supposed to add something to it, as I have already told you. This is clear. So nucleus, then the acrosome is clear. Then we said the centriole, right? That comes and lies into the, the neck region and all. Now here you can see I have made a slight depression. You can see this. I have made a slight depression, right? So that slight depression is actually that neck I am trying to show, but which is usually not so very visible, which is not so very visible. So I'll be showing very lightly over there, okay? And this portion becomes the middle piece. Right? So the centriole which is there, one centriole lies in this depression just below the nucleus, you can see, okay, and the other one comes to lie in this neck region. This is clear, right? So till now what we have made head, so we'll write this over head. This portion is the head. Then very small is that neck portion we have shown, but that later I am marking, that I'll mark it up a little later. Okay, so this, as I said, the dot which we have made, which is where in the depression of that nucleus what we have made, that is the proximal, you can say, the centriole. Okay, so this is the proximal centriole. And what does the proximal centriole do? Proximal centriole, it forms the spindle during the first zygotic division. जब fertilization हो जाएगा और zygote बन जाएगा, zygote will undergo division. So that time spindle formation has to take place. So this proximal centriole will form that spindle during the first zygotic division. So we can add over here that this proximal centriole, what is the function? Spindle formation. Spindle formation during first zygotic division. That is when the zygote is or will undergo the division, right? And the below one, that is called the distal centriole. That is the distal centriole. Right? This distal centriole which is there, right, that actually is going to give rise to this axial filament over here, the exoneme, which will be required, right? So it gives rise to such filaments like this, okay, which forms this axial filament, which is having the 9+2 arrangement. So this is that axial filament. Which is having that 9+2 arrangement. So now if I want to complete this, right, as we said, this if we have to complete, then it will again narrow down from here to form this narrow form of the structure, right? So that is going to form the tail. So this much short portion makes the neck, which is usually not visible. Then this portion makes the middle piece. And from here it is the tail. Right? So the four parts are clear? Right. So which things we have talked about? We talked about the nucleus, the acrosome, the proximal centriole, distal centriole. Yes, what majorly remains is our neighborn cone, right? We said the neighborn cone. Now this as we said, the whole structure is going to get covered with the plasma membrane. So we'll first cover it up with plasma membrane, and then I'm coming back to this Nebenkan. Okay. Right. So this outermost blue membrane which I have made, that is the plasma membrane. I am just labeling it here, okay. This is the plasma membrane, okay. The Nebenkan, that is the spirally arranged ribbon-like structure, which is the mitochondria, which is spirally arranged ribbon-like, that we are calling it as the Nebenkan, right. So if we have to show that, so this red portion that we have made, that indicates that it is the Nebenkan, the spirally arranged mitochondria. So we will label that over here. Right, this is also clear. So that was present where in the middle piece area, correct, and then forms the tail. Now as I said, 2 more membranes I am supposed to add, right, 2 more membranes I am supposed to add. So that is what I am going to do. The first one that I am adding is called galea. Galea is, you can say, a fibrous membrane present on the upper half of the nucleus. Right, so we'll use a different color to indicate. See here. This green that I have made, this portion, and that makes the galea. Okay, galea is a fibrous membrane. But as you see, it covers the upper half only, remaining part is not covered. From the remaining half arises another layer which is called ncert, okay, which is called ncert. Now we'll have to use some other color to show that. Okay, and that covers the other half of the portion, that is also covering the lower half, right. So see from here, so it covers the lower half, galea covers the upper half, remember. right? This is called ncert. Ncert. It is in the lower half, and that is also a membrane. Clear? So this is what actually the structure of human sperm is. We again come to the points back, that it is a haploid, flagellate and motile structure, right? The tail is actually a little longer, but I have not shown that too long over here, right? So the first part, we say the head. The head comprises of the nucleus and the acrosome. So nucleus, as we said, it is a condensed nucleus which contains DNA and the basic proteins, right? And it has the Golgi breaks into two, which was the bigger part was forming acrosome. Acrosome contains what? Remember, sperm lysins. These sperm lysins are nothing but enzymes which help in digesting the egg membrane for the process of fertilization. Alright, so head is formed of the nucleus and the acrosome. Okay, then comes the neck region, which is very, very short, and the two centrioles, one lies into the depression of this nucleus and the other is in the neck region. The upper one, we are calling it proximal centriole. What is the function? It forms the spindle fiber during the first zygotic division, okay? And the distal centriole, it gives rise, right, to the eggshell filament, as we have already discussed. And the distal filament centriole gives rise to the eggshell filament. The eggshell filament has the arrangement of 9+2, the microtubules, right? Then in the middle piece comes the Neberncon, that is the mitochondria, which is spirally arranged. The function is that it provides energy, right, during the movement of the sperm. It has as it has to swim and reach to the egg cell. And then there comes the tail. The whole of the structure is covered by a plasma membrane. There are two other membranes. With the first membrane, which is a fibrous membranes, which covers the upper half part, that is called as galea, and which covers the lower half, that is called the ncert, right? So this is what the structure of human sperm comprises of. Now one more thing which we have to learn here, that is, structure is over, yes, but say in case the human male is heterogametic, as we have said. It has X and Y chromosomes, right? So in males, the genetic makeup is this XY. So when meiosis 1 takes place, meiosis 1, which is the reductional division, they're going to separate, and half of the sperms will have, you can say, the X chromosome, half will have Y, correct? And then the second meiotic division, when gets or takes place, it produces the spermatids, right? So there will be two spermatids with X and two spermatids with Y chromosome, correct? What are these? These are formed are spermatids, correct? And then this spermatids, they are going to undergo spermiogenesis, right? They will undergo what? Spermiogenesis. And so what they will form will be the sperms. So here you can say that half of the sperms will be with X chromosome and half of the sperms will be with that Y chromosome, right? So the sperms which are with X chromosomes, they are they are called as gynos. मैंने ये क्यों बोला बेटा? क्योंकि spell, this is gymnosperms. Understood? So you can say 50% of the sperms will be gymnosperms, कि जिनके पास X chromosome, and 50% of the sperms which will be having the Y chromosome, they are called as androsperms. Right? Gymnosperms and androsperms. This is also clear? Alright. One more thing we need to understand is about a term, viability. What does viability mean? जब ये स्पर्म्स रिलीज होते हैं, प्रोड्यूस हुए हैं, रिलीज हुए हैं, तो कब तक वो इतने स्ट्रॉन्ग है या फर्टिलाइजेशन का पावर उनके पास रहता है, दैट इज एक्चुअली वायबिलिटी. सो द पीरियड फॉर विच यू कैन से द पीरियड द स्पर्म हैज फॉर फर्टिलाइजेशन, कितने टाइम में वो एग को फर्टिलाइज करने का पावर रखता है, कितने टाइम तक, that is viability, right? The period the sperm has for fertilization, the period a sperm has for fertilization, and in humans this period we consider it as 24 hours. So we can say the sperms are viable for 24 hours, right? So I hope all of you are clear with the structure of the human sperm. Once again I have put up the figure and the label so that you can recall, right? And the gymnosperms and androsperms and the viability. Hope you all have followed. Thank you and Jai Bharat.